Hello everyone, welcome back to my channel. This video is specially for first year BDS students preparing for DADH practical exams. Today we will go through some of the most important histological slides, how to identify them step by step, and I have also included my own hand-drawn diagrams and examples to make everything easier to understand and remember. If you haven't watched my previous video on traits of all teeth, do check it out. I have added the link in the description box. So stay tuned till the end. Let's get started. So first we will begin with the chapter cells and stains, and our first slide will be osteoblast and osteocytes. Osteoblast will be arranged in row of cuboidal cells lining the bone surface, and whereas osteocytes will lie in the will be flattened cells present inside the lacunae. This is the lacunae of the bone and within the bone matrix. So this will be the diagram you will draw if the question is on osteoblast and osteocytes. The second slide is on fibroblast. So fibroblast will be seen as spindle shaped. See, these are spindle shaped cells. It will be spindle shaped cell with elongated nucleus and will be in the connective tissue. So this is the connective tissue. You can see bundles of collagen fibers and you can see spindle shaped fibrocytes and fibroblasts. This is how you will draw the slide for fibroblast, and I suggest you need to write an identification feature also over here, which I am suggesting, which will help you gain more marks in exam. For example, for fibroblast you can write spindle shaped cell with elongated nucleus seen in connective tissue, and this will be your diagram. Moving on to next diagram, it is of plasma cells. For plasma cells, this is only the identification feature, that is eccentric placed nucleus with faint basophilic cytoplasm, and this eccentric placed nucleus shows a cartwheel appearance. See, you can see over here, this is a cartwheel appearance of the eccentric placed nucleus. You can see that the nucleus of the cell has been shifted to one side in the slide, and it will be basophilic in appearance. This will be your slide. This will be a diagram to draw for plasma cell, and this will be your identification to write for plasma cells. Next is foreign body type of giant cells. Next is foreign body type giant cells, and in foreign body type of giant cells there is a bigger cell which you can see, and there are multiple nucleus present inside that, and that nucleus are scattered throughout the cytoplasm. So this is only the identification feature for foreign body type of giant cell, and also you will see chronic inflammatory cells, and you need to draw the same in the diagram too, and also the blood vessels. So this will be your diagram for foreign body type of giant cells. Then Langerhans type of giant cells. In Langerhans type of giant cells also the cells will be of bigger size and it will be multinucleated, but the nucleus will be arranged in a horseshoe shaped manner. As you can see, the nucleus is arranged in a horseshoe shaped manner. It won't be scattered throughout the cytoplasm. So this will be the identification feature and the diagram. Next is mast cells. Mast cell is an oval cell with centrally placed nucleus, and there are coarse basophilic granules all around it, or you can say metachromatic granules. This is an identification feature for the mast cells. Next, coming to stains, the most important stain is H&E stain, hematoxylin and eosin stain. So eosin here indicates pink color and hematoxylin indicates purple color, and so you can see the nucleus is stained in a blue or purple color, whereas the cytoplasm is stained in pink color. So this will be your H&E stain. You need to write this in identification feature too. In this there will be a routinely stained H&E slide which won't show anything else. So you need to identify that it is an H&E stain slide. Next is PAS stain. PAS stain will stain the glycogen droplets, the basement membrane and mucopolysaccharides a magenta, that is pinkish purple color, and this will be known as PAS positive. So if in slide you see there is no purplish or bluish shade, then that will be a pass stain, and you can see the glycogen droplets also, that is very prominent. Next is Van Giesen stain. So the Van Giesen stain will stain the collagen fibers red in color and muscles, cytoplasm and other tissues yellow in color. As you can see, this is muscle, this is epithelium, this is yellow in color, whereas collagen fibers is red in color. So this is the identification feature of Van Giesen stain. Next is Mallory stain. Mallory stain will stain the collagen fibers blue in color, the muscles and the cytoplasm red in color, and the nuclei will be prominent red in color, as you can see, and this is used to determine whether the keratin is present or not. So keratin will be stained in red color and pre-keratin layer will stain in orange color. This is the identification feature for Mallory stain. Then Mason's trichome stain. As written, collagen fibers will be stained green or blue in color. The muscle fibers will be stained in red color, and also the keratin if present or cytoplasm if present will be stained in red color, and the nuclei will be stained in black color. So see, this will be the nuclei which will be stained in black color. This is very easy to identify in slide. Everything will be blue or green, the collagen fibers, and there will be muscle fibers which will present in between with red color. Then this is the blood smear. This doesn't come usually, but if it comes, the monocyte will be the largest cell and it will be easy to identify with help of its nucleus. The neutrophil will be multi-lobed, so it will be easy to identify. The eosinophil will be have two nucleus in a headphone shaped manner. The basophil will be having purple or bluish color nucleus or coarse granules which you can see. The lymphocytes is also easy to identify. It will be bluish or purplish in color. Moving on to next is chapter development of tooth. First one is birth stage, which is early birth stage. This doesn't come usually to identify in exams, but if it comes, then this is the diagram which you will draw. This will be the oral epithelium, this will be the oral cavity, and this will be the dental lamina, and it will be the condensed ectomesenchyme below the dental lamina, and this will be the diagram. Next will be birth stage. In birth stage, this comes usually. It will be oral epithelium, then dental lamina over here, then epithelial bud will be seen over here, and there will be condensed ectomesenchymal cells over here. The cells over here will be polygonal in shape, so you need to keep that in mind while drawing the diagram. Moving on to next is cap stage. Now cap stage can come only cap stage also, or cap stage with enamel knot and enamel cord. Usually only cap stage comes. So this will be oral epithelium, this will be dental lamina, this will be outer enamel epithelium, this will be striate reticulum, this will be inner enamel epithelium, this will be dental papilla, and this all will be dental sac. While drawing this diagram, you need to make sure that the shape is cap shape, and if it comes with enamel knot and cord, this part will be extra. Over here you need to make sure that the outer enamel epithelium is made cuboidal shaped cell, and inner enamel epithelium, the cells are made in columnar shape, as that is the difference between these two epitheliums. Moving on to next, the bell stage. In the bell stage, it should look more like a bell shaped. Rest all is same, that is outer oral cavity, oral epithelium, dental lamina. A successional dental lamina will be added in bell stage because that is seen in bell stage. Then outer enamel epithelium with cuboidal shaped cells, inner enamel epithelium with columnar shaped cells, then striate reticulum you need to make, and there will be a stratum intermedium which will be there in between inner enamel epithelium and striate reticulum. then there will be a cervical loop at the meeting of inner enamel epithelium and outer enamel epithelium. Then there will be dental papilla, and this will be dental sac. You just need to make sure that the outer shape of the diagram is bell shaped. Then there will be advanced bell stage, which is early, and advanced bell stage, which is late. Almost this diagram is similar, only just there is difference in the shape of the diagram. So if advanced bell stage comes, you need to draw it like this. That is, oral cavity, oral epithelium will be present, then outer enamel epithelium will be present, but inner enamel epithelium will now be called as ameloblasts. Then there will be stellate reticulum present, stratum intermedium will also be present, and below the ameloblast there will be enamel matrix, which ameloblast will lead to, and then there will be enamel space. This will be dentine, this will be predentine, and this cells will be odontoblast cells, and this dental lamina will turn into developing pulp, and this cervical loop will be present, and a dental sac will be present. Here you need to remember that the dental papilla is turned into the developing pulp over here, and inner enamel epithelium is turned into ameloblast over here. Next, coming to Hertwig's epithelial root sheet. So this diagram is the zoomed version of this. So outer enamel epithelium will be there, cuboidal shape. Then there will be inner enamel epithelium over here, which will be columnar shape. Then there will be stellate reticulum present. Then there will be dentine, predentine, and odontoblast layer also present over here. Then this will be developing pulp area. Then this part will be future CEJ, because from here the root will start to develop. Then it is Hertwig's epithelial root sheet, this part, and this part is of dental sac. This is just a elongated version of this. So this part over here is this part over here, that is dental sac. Moving on to the next chapter, enamel. If enamel rods come as an identification, then you will draw it like this, with a rod sheet present, then cross striation present in between, and this part will be enamel rods. If incremental lines of Retzius comes, you need to draw a diagram like this. For easily remembering the shape of the dentinal tubules, so I don't get confused, I used to draw enamel over here. On the top the crown was there, on the bottom the root was there, so it was like this, and on the right side I used to draw dentine always, so the shape of the dentinal tubules will always follow this pattern, and you won't make mistakes over here. So this will be dentinal tubules, then it will be tenoenamel junction, this will be incremental lines of Retzius, and this will be enamel. Then next is neonatal line. Neonatal line is just an exaggerated incremental line of Retzius, which differentiate the prenatal dentine and postnatal dentine. So the well formed is prenatal dentine, and this is postnatal dentine, which is formed after birth, and this line separates the prenatal with postnatal, that is neonatal line, and the rest remains same, that is this is enamel, this is DEJ, and this is dentine. Moving on to next is gnarled enamel. First of all, what is gnarled enamel? Enamel in which the rods are twisted and interwoven, usually seen on cusp tips or teeth under the microscope. So this is gnarled enamel, which is present on the cusp tip. This is DEJ, dentine enamel junction, and these are dentinal tubules. Moving on to next, Hunter-Schregas bands. First of all, this is visible in longitudinal sections of enamel. This is alternate dark and light bands which is present in the enamel due to change in the direction of enamel rods. So this will be the diagram for Hunter-Schregas bands. Moving on to next, enamel lamellae. What is enamel lamellae? Enamel lamellae is thin leaf-like defect which extends from the surface to the DEJ. This is DEJ. It extends from the surface to the DEJ. It may even cross the DEJ, touch the DEJ, or not even touch. It contains the organic material or air which is visible in longitudinal sections. Moving on to next, enamel tufts. What is enamel tufts? Enamel tufts is small brush-like defects at the dentino-enamel junction, consisting of hypomineralized enamel fibers extending into the enamel. So these are hypomineralized structures. This will be the diagram. You need to see the line of the incremental lines of the ridges and the direction of the dentinal tubules too. Then enamel spindles. Enamel spindles are nothing but the extension of the odontoblastic processes only, which extends into the enamel crossing the dentino-enamel junction. So this will be the identification feature. This will be the enamel. This will be the enamel spindles. This is the lines, incremental lines, and this is the dentine, and this is the DEJ. Over here, the odontoblastic processes are crossing the DEJ. The next is dentino-enamel junction, DEJ. If you can't see anything else other than enamel, dentine, so you can write that as DEJ, or you can write that as enamel or incremental lines of Retzius, or you can write as S-shaped dentinal tubules also, or you can write all the three or four identification features together also, because in that slide you can see all that thing. But if enamel is shown more on the slide and dentine is shown very less on the slide, then it will be related to enamel. But if dentine is shown more, like if this is the slide and over here dentine is shown and only this much is enamel, then it will be related to dentine. But this much of enamel is shown only, and only little bit of dentine can be seen, that it will be related to enamel. You need to remember that. Next is TS of dentinal tubules. So this is intratubular dentine, and this is intertubular dentine, and this is odontoblastic processes which you can see. Next is S-shaped dentinal tubules. As I said, you can mention S-shaped dentinal tubules also if dentine is seen more in the slide. This will be the diagram for that. Then Y-shaped dentinal tubules. This you can identify by a Y-shaped, prominent many Y-shaped tubules will be seen over at the top, and this tubules will branch near the dentino-enamel junction only, and not further, not over here. It will branch over here only. Then next is incremental lines of von Ebner. As there is incremental lines of Retzius in enamel, there is incremental lines of von Ebner in dentine. So if dentine is seen in more quantity, you can write this part. Then next is interglobular dentine. So what is interglobular dentine? Interglobular dentine is hypomineralized structures which is present between the globules. So see over here, if this and this are globules, this is the hypomineralized structures which is present between the globules and which is irregular in shape and not mineralized. This is usually formed between mental dentine and circumpulpal dentine, just below the mental dentine. Moving on to next, Tomes granular layer. So over here, this is the Tomes granular layer. It is an hypomineralized layer of dentine in the cervical root region. So this over here enamel ends, and over here the root starts. So this is present in the root region and just beneath the cementum. That is over here it is cementum and just present beneath the cementum. So this is the Tomes granular layer. Then dead tracts. It is seen different in transmitted light and seen different in reflected light. In transmitted light, it is seen very dark in appearance. What is dead tracts? Dead tracts is empty dentinal tubules which in which odontoblastic processes are not present. So this is seen dark in transmitted light and light in reflected light. It is seen empty space over here. This is the dead tract in reflected light. Rest you can see that enamel is present over here, and this is dentine, and this is the pulp cavity. Same over here, enamel is present, this is dentine, and this is the pulp cavity. Moving on to next, secondary dentine. What is secondary dentine? Secondary dentine is formed after the tooth eruption as the normal physiological process, and it is usually less tubular and darker. As you can see that this is darker than the primary dentine. So this is the primary dentine, and this is the secondary dentine which is formed after the tooth is erupt. erupted, and this is darker in color than the primary dentine. You have to mention the pre-dentine also in this, and the odontoplastic processes also, and the dentinal pulp also. Moving on to next, transparent dentine. This occurs usually with response to age or to an external stimuli. This is sclerotic dentine with obliterated dentinal tubules which appear translucent under the microscope. As you can see over here, it appears translucent and it appears to be empty, like white appearance, and transparent dentine. Therefore, this is known as transparent dentine. As you can see, this is the root canal, this is the radicular dentine. Over here, the dentine is not visible. So, this is the transparent dentine, and this is the layer of the cementum. Moving on to next, zones of pulp. This is the next chapter, that is pulp. So, zones of pulp is easy. There is primary dentine, followed by secondary dentine. Then there is pre-dentine with followed by odontoblastic layer. Then there is cell-free zone. Over here, there is no cells present. Then there is cell-rich zone, that are many cells are present in this zone, and there is pulp core which is present beneath. Usually, the zones of pulp don't come. Along with that, attached pulp stone or free pulp stones usually comes in spotters. So this is free pulp stones. You can see that the stones are on the pulp core. So this is discrete calcified masses which is entirely located in the pulp tissue. That's why it is known as pulp stone. If it is attached to the layer of odontoplastic and dentine, then it will be attached pulp stone, and if it is completely embedded into the dentine, it will be known as embedded pulp stones. Rest all will remain the same, that is primary dentine, secondary dentine, pre-dentine, odontoplastic layer, and pulp core. These area of the stone will differ, that is free pulp stone in the pulp core, attached pulp stone, and embedded pulp stone in the dentine. Moving on to next, linear calcification. So linear calcification is calcified deposits that appear as thin. You can see that it is thin and it is elongated. Elongated lines along the blood vessels. This is blood vessels or collagen fibers in the dental pulp. So this is pulp. You can see the elongated fibers, and this is primary dentine. This is secondary dentine. Moving on to next, zones of cementum. We move to next chapter, cementum. Zones of cementum is also not usually asked question. Zones of cementum, as you can see, I have labeled that this is dentine, this is cementum, then there is a layer of cementoid, and then there is cementoblast, then there is PDL, this is periodontal, that is periodontal ligament, and here you can see alveolar bone. This is not usually asked slide. Then acellular cementum. As you can see, there are no cells present in this cementum. So you can see this is enamel, this is cemento-enamel junction, this is dentine, this is acellular cementum. Then beneath the cementum there is always present a Tomes layer, granular layer, and there is Sharpie's fibers also you can see. The acellular cementum is usually present near the cervical region of the root. Then there is cellular cementum. This is usually present near the apical region of the root. This will be just similar to the acellular cementum, but there will be addition of cells, that is cementocytes. That's why it is known as cellular cementum. Moving on to next, Sharpie's fibers. These Sharpie's fibers are actually the terminal ends of the periodontal ligament which is present beside that, which helps in attachment of cementum and alveolar bone. So these are those terminal ends of the periodontal ligament fibers, that is known as Sharpie's fibers when entered into the cementum. So this will be the incremental lines of Salter, Sharpie's fiber, and then Tomes granular layer, and there will be dentine. Then incremental lines of Salter, as there was incremental lines of Retzius and incremental lines of von Ebner in enamel and dentine, in cementum it is incremental lines of Salter. If you can see nothing, you can write incremental lines of Salter or Tomes granular layer also. For this you can write. Then there is cemento-enamel junction. These are of three types, that is overlap, edge to edge, and gap junction. The overlap junction will overlap the enamel which is present above it. The edge to edge will just touch the enamel which is present above it, and the gap junction there will be a little bit exposed dentine, as cementum will start at some distance away from the enamel. You can also mention that overlap type of junction is in most of the population, that is 60% of population. Then H to H junction is seen in 30% of population, and it is also known as butt joint. And gap junction is seen in only 10% of the population, which exposes the dentine. These are the identification feature you can write. Then there is hypercementosis, that is more layer of cementum is formed near the apical portion of the root. So this will be the root canal, radicular dentine, cementum, and it is hypercementosis. So just instead of this, it is more an irregular shape, and there will be more cementocytes also present in this layer. Moving on to next, gingival and transeptal fibers. We move to the next chapter, that is periodontal ligament. So as you can see in this diagram, that this is the interdental bone, that is alveolar bone. Over here it is the dentine or the teeth of the two adjacent teeth. This is first and this is second, for example, and this is bone present between the two teeth. So this will be the enamel of first teeth and this will be enamel of the second teeth. There will be gingival epithelium between the two teeth, and this is the dentine enamel junction, as this is dentine and this is enamel. Then there is cemental enamel junction also over here and here. Then this is dentine, this is cementum, this line is cementum. Then there is gingival fibers from here to here, and transeptal fibers from here to here, that is fibers that extend from the cementum of one tooth. This is for one tooth to the cementum of the adjacent tooth. This is second tooth, extends from first tooth to second tooth over the alveolar bone, and it helps to maintain the teeth in alignment. Then as you can see, over here there are horizontal fibers of PDL, and over here there is crestal fibers, or also known as apical fibers of periodontal ligament. Moving on to next, alveolar crestal horizontal or oblique fibers. As I have mentioned now only in the above diagram, this is the zoomed version of this part. See, as you can see, this is just elongated over here. Over here there were transeptal fibers over here, so you can see that transeptal fibers are present over here. Then there is apical fibers, crestal fibers. Then here there is horizontal fibers, and below that there are oblique fibers. The horizontal fibers turn to oblique fibers below. Then apical fibers. Apical fibers are present just beneath the apex of the teeth. Like this is the apical fibers, this is the alveolar bone, this is the teeth, this is the cementum, and this is the root of the teeth, and this is the PDL. Over here there is oblique fibers, and over here there are apical fibers. Then interradicular fibers. These fibers are present between two roots of the same teeth. Transeptal were between two adjacent teeth, and this is two roots of the same teeth, interradicular fibers. As you can see, if it is a molar teeth, this is the first root, this is the second root, and these fibers between the two roots of the teeth, these are known as interradicular fibers. Rest all you can see the labeling if you want to. These are easy, so I am skipping that part. Moving on to next, free cementicles and embedded cementicles. First of all, what is cementicle? Cementicle is small, round or oval calcified bodies in the dental pulp or periodontal ligament, which is either free or attached or embedded. We just need to study the free and the attached one, the embedded one. So this is the calcified bodies present in the PDL. So over here you can see this is the dentine, this is the pulp canal, this is the cementum, this entire thing is the PDL space, and this is the calcified masses of cementicles. This is the alveolar bone. This is present freely, so this is free cementicles. This is embedded into the cementum, that's why this is embedded cementicles. Moving on to next, epithelial cell rest of Malleus. So this epithelial cell rest of Malleus are the residual epithelial cells from the Hertwig's epithelial root sheet located in the PDL often forming clusters or strands. This, as over here you can see that these are in clusters. Moving on to next, now we move to the next topic, OMM, oral mucous membrane, and first one is gingiva. Gingiva is parakeratinized epithelium that, as you can see, that it is stratified squamous epithelium. Then there is lamina dura present. There is long slender rete ridges which are numerous in number. As you can see, there are many rete ridges present, and this is parakeratinized epithelium with pycnotic nucleus present. That's why it is known as parakeratinized. This is the peculiar feature of gingiva, that is long and slender rete ridges and parakeratinized epithelium. Moving on to next, hard palate. Hard palate has two parts, anterolateral zone and posterolateral zone. In anterolateral zone, the submucosa shows adipose tissue, whereas in posterolateral zone, the submucosa shows a mucus acini. A trick to remember that is anterolateral. So A. Anterolateral will show adipose tissue, whereas posterolateral will show mucus cells. Then the rete ridges is broad and blunt. As you can see, it is not pointed like it was over here. This was slender and there were many rete ridges. Over here there is only 1, 2, 3 and 4 rete ridges, which is broad and blunt, and the keratin layer is orthokeratinized, and there is prominent granular cell layer which is present beneath the orthokeratinized layer. In orthokeratinized layer, the pycnotic nucleus are not present. So as you can see that this is the stratified squamous epithelium, then this is lamina propria layer, and this is submucosal layer. Over here everything is same as that in anterior lateral zone, just the submucosa is replaced from mucus acini cells. Moving on to next, buccal mucosa. In buccal mucosa also there is thick bulbous rete ridges. It is not long and slender like in gingiva. Then it doesn't has an keratin layer, that is, it is non-keratinized stratified squamous epithelium. Non-keratinized, no keratin layer is present above the epithelium. Then there are thin and short irregular connective tissue papillae, as you can see over here. Then the lamina propria shows loose connective tissue, and the submucosa shows an adipose tissue, connective tissue. This is the submucosa which shows adipose connective tissue, and mixed salivary glands are present. Also you can see the cut sections of the muscle tissues. Then buccal mucosa showing Fordyce spots. What is Fordyce spots? It is an ectopic sebaceous glands in the oral mucosa. Over here rest features will be of buccal mucosa only, that is, it is non-keratinized. Then thick and bulbous rete ridges, thin, short and irregular connective tissue papillae. Then lamina dura shows loose connective tissue. This is the part of the sebaceous gland, and rest part like mixed salivary gland and muscle tissue will remain the same. Just this part will add up, that is the sebaceous gland, which shows the Fordyce spot. Next is fungiform and filiform papillae. So the filiform papillae will be keratinized stratified squamous epithelium, whereas the fungiform papillae will be non-keratinized stratified squamous epithelium. As you can see, there is no keratin layer present in this, and there is keratin layer present in this. So this is keratinized and this is non-keratinized, and you can see there is presence of taste bud also in the fungiform papilla, and beneath you can see muscle fibers which are longitudinally and cross-sectionally cut, that is haphazardly cut muscle fibers. Then circumvallate papillae. In this, the lateral wall will be non-keratinized squamous epithelium, but above there will be presence of keratinized stratified squamous epithelium. Then there will be presence of taste buds on the lateral walls of the circumvallate papilla. Also you can see salivary glands like von Ebner salivary glands and muscle fibers. Especially the von Ebner salivary glands are present near the circumvallate papilla, because in the circumvallate papilla the taste buds are present in the lateral walls, and the von Ebner salivary gland secretes a serous secretion. So it is easier that watery secretion, Serous secretion is basically a watery secretion which is easier to drain over here and clean this part of area where taste buds are present. Then the next diagram is of taste buds. You can see a taste spore, then there is stratified squamous epithelium, and these are the taste cells and supporting cells, and this is the connective tissue. This is also easy to identify a taste bud. Then there is a slide of the lip. In the lip there is labial mucosa which is non-keratinized squamous epithelium. Then there is vomular zone or transitional zone or red zone, and there is skin which is orthokeratinized stratified squamous epithelium. There over here there is long connective tissue papilla with capillaries. Then there is hair follicle also present near the skin area. Then there is muscles present over here, sebaceous glands present over here. Then there are sweat glands and mucus glands also present in the slide of the lip. This is a little bit complicated slide. Then slide of the junctional epithelium. In the junctional epithelium there is free gingiva which is keratinized. Then there is attached gingiva which is also keratinized. Then there is alveolar mucosa which is non-keratinized, so you need to draw keratin till here only. This is the mucogingival junction which separates attached gingiva with alveolar mucosa. This is the sulcus, dentine, enamel, then sulcular mucosa which is non-keratinized. Then there is free gingival groove which separates free gingiva with attached gingiva, and this is the junctional epithelium over here. Rest all like cementum, PDL and alveolar bone is also present. Moving on to next chapter which is salivary glands, and the first slide is serous salivary gland. In serous salivary glands and in this chapter you can draw the enlarged structures of the glands also, like I have drawn over here of serous salivary gland, in which there is pyramidal shaped acinar cell with spherical nucleus. You can see that this is spherical nucleus. Then apically placed eosinophilic zymogen granules. As you can see, I have drawn over here eosinophilic zymogen granules and epithelioid cells also in the enlarged diagram, in the inlet diagram. And in the normal diagram you will draw interlobular duct, serous acini, multiple serous acini. Then there will be intralobular duct, the connective tissue and the adipose tissue also present. This drawing this inlet diagram is very important for salivary glands chapter. Moving on to next, mucous salivary glands. In mucous salivary glands there will be pyramidal shaped mucous acini cells present. There will be flat nucleus which is basally placed. As you can see over here, this nucleus is basally placed, and there will be myepithelial cells also present. In this normal diagram you will draw interlobular duct, the mucous acini, the intralobular duct and the connective tissue, same as the serous salivary glands, just the serous acini will get replaced by the mucous acini. In the mixed salivary glands you will draw both the mucous acini and the serous acini, and also there will be mucous acini with serous demilune present. As you can see over here, this is mucous acini with presence of serous demilune. Rest everything will remain same. Moving on to next, alveolar bone chapter. The first is compact bone. In this you can see Haversial canal at the center. Then there is concentrically arranged lamella. Then between the concentric arranged lamella, that in between there is interstitial lamella and osteocytes present throughout the concentric lamella. So this entire unit is known as an osteon, which contains a central canal, the concentric lamellas and the lacunae with the osteocytes and the canaliculi present. Moving on to next, the trabeculated bone. So this is an cancellous bone with interconnected trabeculae connecting osteocytes in the lacunae and surrounded by the bone marrow spaces. As you can see, these are the trabeculae of the lamellated bone. Here you can see a fatty marrow and hemopoietic marrow also present, and these are the osteocytes which are present inside the lacunae. This is the trabeculated or spongy or cancellous bone. Then there is immature bone, also known as Bowman bone. Over here the osteocytes are randomly located inside the lacunae, and collagen fibers are irregularly and randomly arranged over here, as you can see. These are the osteoblasts. The layer, this layer is known as osteoid layer. The cells inside this are osteocytes. Then this, you can see that it is patchy basophilic part of the immature bone or the woven bone. This is the connective tissue with collagens randomly arranged. Moving on to next, maxillary sinus lining. In this you can see that there is presence of pseudostratified ciliated columnar epithelium with goblet cells present, and there will be cilia as it is ciliated columnar epithelium. It will give an appearance of striation, that is multiple cells present, but there will be only a single layer of cells present which are elongated like this. In this there will be presence of mucous salivary acini also and serous salivary acini also. So this is the identification feature, that is pseudostratified ciliated columnar epithelium overlying a thin connective tissue. This was the last slide with my own hand-drawn diagrams. Now we will proceed with some of the examples from which you will be able to identify in exam that which slide the particular represents. You can pause the video and try to identify by yourself also that what the particular slide represents. So this is the first slide. Over here you can see that there is long slender rete present, and over here there is a layer of keratin also. So this slide is of gingiva. Moving on to next, this is very easy slide. As you can see, there is yellow stain and red stain. So this is Van Giesen stain, as the red is stained by the collagen fibers and yellow is stained by the epithelium and other connective tissues. Moving on to next, this is Mallory stain. As you can see, the collagen fibers are stained blue in color. Then there is layer of keratin also present with a red color containing keratin and orange layer containing a pre-keratin layer. And if the photo was more clear, you can see the red spots also of connective tissue like blood vessels, and over here also the nucleus of the stratified squamous epithelium. So this will be Mallory stain. Moving on to next slide. If you carefully see, over here there are odontoblastic layers originating from the dentine into the enamel. So these are the spindle fibers from dentine to the enamel. So this will be the spindle fibers. Moving on to next. This doesn't show a particular thing, but you can write that this is dentino-enamel junction also, or you can write incremental lines of enamel also. As you can see over here, enamel is more and dentine is less. So this will be related something to enamel. So you can write dentino-enamel junction also or incremental lines of Retzius also. But in diagram you need to mention everything, like this is dentino-enamel junction, this is incremental lines of Retzius, this is enamel, this is dentine, this is S-shaped dentinal tubules, everything. This is just crack present in the tooth. This is not a lamella. As you can see, this is the fractured tooth over here. This. The next, this is dental lamella. Moving on to next slide. As you can see, there are giant cells present. This, all these are giant cells. So this is foreign body type of giant cells, and this is not Langerhans type. This is foreign body type, as the nucleus is haphazardly arranged. Moving on to our next slide. This is a slide of fibroblast. As you can see, there are fibroblasts present in this. So this is the slide of fibroblasts. Moving on to next, this is an cap stage. As you can see, this is an cap shape appearance. This is not bell, completely bell. This is cap stage, as only this much is formed. So this will be cap stage of development of tooth. Then this will be our advanced bell stage. As you can see, the tooth is almost developed. The shape of the tooth has almost come. So this is advanced Bell stage. Then moving on to next, this is bird stage. As you can see, this is the oral epithelium and this is the bird which you can see. This is the bird which you can see. Moving on to next, this is Bell stage. As you can see, this is bell shape and there was difference in cap stage. See, this is the cap stage where shape is resembling like a cap, whereas this is a bell stage. You can see properly that it is showing a bell stage. Moving on to next, this slide shows interglobular dentine. As you can see, there are hypomineralized structures present. This is interglobular dentine. This slide shows dead tracts. These are dead tracts which contains dead odontoblastic processes. And these are dead tracts in transmitted light, because in transmitted light it is seen black and in reflected light it is seen white. Moving on to next, this you can write acellular cementum also. As you can see, this is acellular cementum and just beneath the acellular cementum this layer is Tomes granular layer. So you can write Tomes granular layer also or acellular cementum also. This part is dentine, as you can see. Then this is cellular cementum. As you can see, there are cells present in the cementum which are cementocytes. This is a slight granular layer and this is the dentine. So these are the cells, cementocytes, which are present in the cellular cementum. Moving on to next, this is cemento-enamel junction. You can see over here it is getting overlapped. So this is overlap type. Over here there is gap. This is the gap. This is the cementum and this is the enamel. So this is the gap where dentine is exposed. So this is gap type CEJ and this is edge to edge type. Just it is touching the enamel. The cementum and the enamel are just touching. This is edge to edge type of CEJ. That's it for today's video on histological slide identification. I hope the hand-drawn diagrams and the examples which I took will make it easier for you to recognize slides in your practical exams. If you haven't watched my traits of all teeth video yet, the link is in the description box. Do check that out. That video is also great for practical exams. If you found this video helpful, like, share and subscribe my channel for more exam focused BDS practical videos or lecture videos. Do share it with your batchmates and friends and make it easier for them too. Stay tuned for the next video in the DADH practical series. Best of luck for your exams. Thank you. Bye bye.